Hello, in this video we're going to continue in chapter 7. We're focusing on how molecules move across membranes, and we're on question 5 of our PowerPoint. And the question is, how does facilitated diffusion differ from simple diffusion? Explain the difference between a channel and a carrier protein. Okay, diffusion, remember, is a passive process, so no energy is required for either of them. But diffusion involves the movement of molecules from high to low concentration, right? There are two different types of diffusion. One I call simple diffusion, which doesn't need a helper. Simple diffusion, molecules can simply diffuse directly across a membrane. Oxygen can diffuse directly across the membrane without a helper. And then facilitated diffusion. Facilitated requires a protein helper. That's the big difference. A protein helper that sits in the membrane that allows the molecule to cross the membrane, to diffuse down its concentration gradient. There are different kinds of protein helpers. One's called a channel protein. The other is called a carrier protein. Let's take a look at a diagram from our textbook that shows this. So a channel protein is shown on the top diagram. In a channel protein, the molecules here are moving from high to low concentration through the channel protein. The channel protein is a transporter that doesn't change shape. It allows maybe one or maybe two specific molecules to pass through. It doesn't allow just anything to pass through, but it doesn't change shape. A good example of that would be aquaporins. This is just an example, okay? In this diagram on the bottom, we've got a carrier protein. A carrier protein actually picks up a molecule and transports it across the membrane down its concentration gradient again from high to low concentration, but it changes shape along the way. The carrier protein actually changes shape. Is facilitated diffusion a passive process? Put a big fat yes. Yes, it is. It doesn't require energy. Molecules are still diffusing. They're still moving down their concentration gradients. Okay. Let's take a look at question 6. Question 6. What are the major differences between active transport, there's our new one here, and facilitated diffusion? Think about whether molecules move down their concentration gradients, whether a transport protein is required, whether energy is required. Okay, so facilitated diffusion, we're pretty confident we know what that is. A good example would be aquaporins in the diffusion of water, or the diffusion of water is osmosis, right? So in facilitated diffusion, which way do molecules move? They move from high to low concentration gradient, right? They move down their concentration gradient, or with the gradient. They spread out. Is a transport protein required? Yep. That's why it's called facilitated. And is energy required? Nope. It's diffusion. It's passive. What about active transport? Active transport, it has the word active in it. So, okay, this is not going to be passive. And so right away, this is how we know it's different. In this case, then yes, energy is required. It's active. We're pumping molecules across the membrane and we're moving them against their concentration gradients. From low to high concentrations, we're moving them against their concentration gradients. We'll take a look at some examples of that. And is a transport protein required? Yes, we're going to need a protein to help pump them across the membranes. And I really like the term, think about this as a pump. Pumping the molecule across the membrane. Let's look at a couple of examples of active transport. A couple pictures. Here's a picture of a lysosome. Lysosome is from chapter 6, right? Remember, it's a little tiny sac in cells that is a digestive sac or a recycling sac where you've got a whole bunch of different hydrolytic enzymes at a very low pH. which means is a high concentration of protons inside the lysosomes relative to outside in the cytosol. Okay? Lots of protons in here. How do those protons get in here? Dun-da-da. Welcome to active transport. In this picture, we've got a picture of active transport happening. Okay, protons are being pumped across the membrane through a transport protein, shown there in blue, and that requires ATP. So ATP is being broken down into its components as protons are being pumped into this lysosome. That's a nice example of active transport. Here's another picture from our textbook that you can compare and contrast both passive and active transport. The kicker here is this active transport is a little bit complicated. This protein that's involved in active transport here, it's transporting two different types of molecules, but they're both going against their concentration gradients. So we've got these diamonds that are being pumped up or across the membrane, and we've got these circles that are in high concentration up here, and they're being pumped. I'm sorry, I said that the wrong way. In this case, the circles are being pumped across the membrane as well. That seems odd. Wow, that's a weird diagram. Okay, let's not worry about that one so much. In this case, what we're doing is I said it backwards. We're pushing or pumping these diamonds against their concentration gradient. We're concentrating these diamonds over here, and we're pumping the circles against their concentration gradient and concentrating them over here. Sorry that I confused you. Okay, now we've got a very complex question, but don't worry, it's not that complicated. What I want you to do is go to chapter 9 in our textbook. We're going to be focusing on cellular respiration in chapter 9. You know nothing about it, that's okay. But there's a picture in the chapter that I want you to take a look at, and I'm going to show it to you in a second. And the question that I have for you is, are the hydrogen ions, we would call those protons, traveling through ATP synthase protein by active transport or facilitated diffusion? All right, let's go take a look at this and try to answer that question. We're going to spend tons of time in chapter 9, but I wanted to just give you a quick preview of it because it's kind of cool. In chapter 9, we're going to focus on mitochondria, and this membrane is a membrane